Initially we just added data quality in a paragraph only. Later what I did, I created a table for it.
Yeah.
So it shows that how many total sequences were there and what
Yeah.
was the quality. The entire theme of doing data quality is to analyze whatever DNA we extracted is up to the optimal level so that we can process it.
Yeah. So when this data, the way the numbers and all this is put, how is that suppose 96.60 for S6 you have put, and the total input sequence you have shown as 18,309. So how do I say, like, how do I, that sequence total number is that much?
No. It's just that we added the first, how much total sequences were added first, which is total input.
Okay.
And then we keep, how we analyze is we first add data, add sequences, and then we see how many sequences can be read. If some of the sequences are shorter, after cutting it, it's discarded. Then the median sequence length we consider is around whatever base pair is given. So we consider that, and then we see how much sequence is retained after filtering. It's essentially a filtrating system to read whether the amount of input we have added, now how much out of that amount we can actually read. So percentage is showing that suppose we added 100%, out of this 97.997% of data is readable, can be used further.
Okay.
Yeah.
Okay.
That I have written very clearly.
Okay. Yeah.
Then we come to OUT picking. It's operational taxonomic unit.
Yeah.
So it's a pipeline, microbiome analysis pipeline. It has multiple parts, so we need not to go with everything. So it's essentially a pipeline to analyze now the data. Suppose 97.97% we have. Now it's time to analyze. So table 2 shows how it's analyzed. So suppose that much, how much, 1700 something was remained.
Yeah.
So that sequence, wait, let me go. I forgot the total sequence remained 97,530, out of which now we are reading in a linear manner. So how many reads were done? So for S10, 3,570 reads done. So out of which 468 OTUs were observed. So 468 OTUs means that many sequences in linear manner. Now we can use it as that many units can be used as to analyze microbiome, to analyze further and see which sequence matches with which particular microbiome according to genus and all.
Okay, so do you have some standard with which you match, or how do you do that?
Okay, so the softwares we use, they come with preset standards.
Okay.
So that is used, and if you can see, I have added interpretations also.
Yeah.
So lowest read count but moderate OTU count means even though it's the lowest read, it still comes with moderate OTUs. So lowest read means 3,572 out of 17,000 is the lowest that the software can detect, but still the OTU, the operational taxonomic unit, is still moderate.
Okay.
So it shows that there is a microbial diversity and it can be further utilized.
Okay. Yeah.
And of course, highest OTU count means there is microbial diversity, it can be counted.
So that means when there will be low read count and low OTU or high OTU, that will signify
lowest read count and lowest OTU count means there is a possibility that the microbial diversity is really less and it might be difficult to read.
Okay.
So in those scenarios, we have to start from starting, like DNA sample collection, DNA isolation, and redo it.
Okay.
So if you check, there is moderate read count and lower OTU, still readable. Then highest, lowest OTU, still readable.
Yeah.
Okay.
Yeah.
So this is the statistical
right.
term.
Right. Yeah. Got it.
The table 2 is reads an ODU outline, and then all of this data is statistically analyzed also.
Yeah.
Okay, so
Yeah.
all, because we have 5 samples, so we want
Right.
to see how statistically significant all these things are.
Yeah.
So if you see the total across samples, total reads, then total ODU, then interpretation,
Yeah.
which was the minimum reads, which was the minimum ODU, which was the maximum. So this is all statistical analysis. If needed, you can show. If
Yeah.
not, it just proves that whatever data we are using, it's statistically significant.
Yeah. Okay. Okay.
So all that statistical is I have written key points.
Yeah.
S11 shows highest microbial diversity. Then S7 most read but lowest ODUs, suggesting microbial community dominated by few species. It just
Okay.
means that one of the species is
Yeah.
very dominant in
Yes.
that
Yes. Got it. Yeah.
yeah. So I think that much is
That is
clear,
okay.
right?
Yeah. Yeah. Now NSTI onwards. Yeah.
Yeah, it's the nearest sequence taxon index. So I told you, right, there is a microbiome analysis
Yes.
pipeline. So
Huh.
these are the different analysis we do.
Hmm.
Okay, so again, in that analysis is the nearest sequence taxon index. It shows that okay, how to explain? Okay, each organism or each live microorganism is categorized.
Huh.
Into, so they are categorized into kingdoms, which is essentially like animal, plant,
Yeah. Got it.
okay.
Yeah.
So there is like genus, species,
Yeah.
and
Phylum and all. Yeah.
yeah. So we have to find that this sequence is nearest to what.
Yeah. Okay.
So it NSTI do that.
Okay.
It have a huge database, so we match with it. So then we go for.
So in this, are you able to segregate in terms of like whether it's a eukaryote or prokaryote or archaea?
Not in this. This is just core analysis. The
Acha.
next after this we go to taxonomy.
Okay.
Okay. So for example, S10. Okay. So okay. Okay. So what we NSTI is just shows that how much taxonomic similarity is there between identified microbial species. Suppose one sample have multiple microbiome. So it might be that they are closely related, so the values will vary, and it might be that they are very different. They belong to very different taxonomic or their own lineages. So the values might vary. So if you look at it, there is a table. This table is automatically generated. It shows the values and the what you say metric weighted NSTI.
Yeah.
Okay. So
now just hold on, because see, now suppose these are the samples collected from the 4 different sites of the same water body. Okay, now there won't be much difference. If suppose I go from one water body to other, there will be a lot of difference, but if it is in the same system, maybe it will not be so much different.
Not necessarily. Water is something which flows.
Yeah.
So wherever.
No, I'm talking about the lake system. Suppose it's a lake system, because we are right now talking about the lake system.
Lake is a huge system.
Yeah.
Lake have different. If it's a circular lake, then there is different points of location.
Yeah.
We can accumulate it and then generate a cumulative microbial profile, or location we pick north of lake. So north of lake have this. It might not be very stable because the water moves, but still it gives the overall idea what might be the microbiome profile in that particular zone.
Okay, so but the data wise, the data which is seen, just go up little. The data which is there like 0.22 I can see, and above that is suppose 0.142. So how I interpret that?
Okay, let me go to
the values. By values I'm talking about. Yeah.
If you go to that was the table, then
okay, yeah.
Individual scoring.
Yeah.
So 0.244 is moderately high, suggesting some well-characterized species like Cornelia and Leptospirea.
Okay.
Then I have written.
Yeah, got it. Now got it. Yeah.
Okay. So and then the interpretation is also written.
Yes.
Just in case it's not clear.
Yes.
then key takeaways.
Yeah.
Again, it's to just to give a better idea.
Yeah.
Okay, now we come to taxonomic classification.
Right.
For taxonomic classification, there is a huge folder.
Yeah.
So what we do, we plotted bar plot summary and then Krona. Krona is sample wise. Bar plot analysis is accumulating them into phylum, classes, family, like that. Okay. So this is a bar plot of phylum level.
Yeah, but then the color wise, this color when it is there, how do I mean, like how they signify like colors?
Oh.
Because those bar plots, I was thinking why it is green, why it is red. So they signify for different kind of.
There is like thousands of phylum. So if the color is something, we are provided a gradient, which gradient you want to select. So individually identifying each color is difficult. That's why we give percentage, how much percentage of proteobacteria is. So not necessarily these colors can change. It's not something very fixed. It's a gradient provided by the software.
Okay, but then because normally we see that suppose this red is that center one, that is I think S7, it is showing a lot of red. So maybe that stands for some particular genus or species or something like that.
It might be proteobacteria. Let me. I think I have the bar plot also.
Yeah, because that color wise also, if it would have written no, like blue stands for this, red stands for this, something of that sort. So probably.
We do have a file for that, but again, it's a huge file. So,
so at least one, that's why I said that at least one if we can give a proper thing instead of all the samples. So.
No, I can provide you this because the software generates it. But okay, what I can do, the highest color range. Suppose in this you can see blue, red, the blue, the yellow, and I think this green. Or I don't know if it's called even green. I will make small remarks under every bar plot. Yeah. That this red signifies this and this.
Yeah.
Okay. So for every bar plot, I have written the phylum, what is the key. This is the percentage generated by the software.
Yeah, but then here it is on the basis of phylum. Because I saw that phylum class wise, and that's how you have gone.
Basis of phylum, yeah. Then after phylum, you will also find class level distribution.
Yeah.
Same, I will do that. Make for every bar plot. Just provide me a half a day.
Yeah.
I can do that.
Yeah.
You can see when we go to even order level, now the color difference
change. Yeah. So that's what I was thinking. Yeah.
Because as we descend into from
more specification, as you are going for more specific.
The number of order is more than number of phylum. So of course the bacteria's distribution is going to be overall.
Yeah.
That's why every percentage. But I got your point. If you want to define some
right,
okay, that I can do. This is the bar plot summary. I think you get it.
Yeah.
The Krona plot is individual samples.
Right.
So this entire plot shows every microbe, at least the genus level, how much percentage of that genus is present in that particular sample. It's just that much.
So this is at the genus level it is shown, but is it written somewhere that it's a Krona plot shows about the genus?
I think I have written. Let me check.
Go up so that we can make out. Yeah. It's not.
No, because the Krona plot is because species wise is very difficult to visualize. So no, actually this can be shown. Okay, see, bacteria, proteobacteria, or do you want me to
zoom
name them? Which is the species, which is the genus, which is the okay? That might be much more useful. I don't think I can edit
that is why I said at least one. If we can make it more detailed instead of all, that would give us much more good picture.
No, the closer circles are phylum, then family, order. S7 is sample, bacteria is kingdom, then proteobacteria is phylum, bacterio is phylum, this is phylum, this. It's grow, like the closer to center is the overall larger umbrella. The more the corona plot is spreading, it goes from order, class, family, genus. So at the end, most of them are genus level, because it's difficult to identify species level. It's very tricky in this situation.
Yeah, and in this you have marked that with blue and grey, this on the side, that with archaea, and I think what is that down one, bacteria?
Other which is not identified. Archaea is 0%.
Okay.
Other S5, it might be fungi or it might be microalgae.
Acha.
Those are not identified in 16S.
Okay.
So it shows 0.01.
So this is with the help of 16S that it has been plotted. Okay. Please drink little water.
Yeah. Okay, what I can do is, I think I made a table also. Let me go. See, in sample 10, there is archaea also. So bacteria and archaea are present, which is identified in this. So it's plotted.
So this is which number sample? This is Varal Devi Lake soil. Figure 10 it's showing, no, down written. It's the soil sample.
Sample number 11, Varal Devi Lake softening site soil.
So it's a soil sample.
Yeah.
So I think in the water samples it's not shown, and in the sediment we are getting this more clear.
Yeah, it might be. Plus, these archaeas which are mentioned here, they have similarity. It's essentially a very close relative to bacteria, but they have certain
I know
similarities.
That. Yeah, that pathogenic this thing is lesser in them. So
yeah. So whatever is picked up, it picked up. Whatever doesn't show, it doesn't show. Okay. See, so every, I think, summary of corona plot I have given, which dominant phylum is there, which dominant genera is also there.
Right.
So in the table format, because editing those pictures is difficult. Those are software generated.
Yeah.
So interpretation and everything is given down.
So can I also get a little bit of interpretation here? If suppose a certain kind of bacteria is present there, so what may be the reason of that presence of that particular bacteria? Like
yeah, I think I have added that. So so this is the statistical analysis. I haven't added because it won't help you in this.
But why you say statistical analysis won't help?
Because you are trying to present that this is the microbial diversity. Even if you say that Porphyry or Chrysopoli plot says it's 0.1 significance or the value is 1, it's a huge number of data, and making someone understand the biostat part of might be really, really tricky. So to present in a very comprehensive manner, I would suggest to skip just by saying that statistically the data was good is enough for statistical analysis.
That is okay, but if suppose I want to use it for some publication,
I can provide you the data. The statistical analysis is also very huge, and I don't know how to even write it in a publication. Most of the time when we are publishing for metagenomics data, we usually skip statistical analysis or write in 1-2 line. That's it.
Okay, but then mainly in other cases, like in other subjects or other way, when they, like suppose in botany, zoology and all, they go more on a statistical mode.
The amount of data is very less.
Too high. Achha,
Here
okay.
the amount of data is millions.
Yeah, true.
So writing 1-2 line suggests.
No, that is why I said that instead of going for all, at least one case, if we can do it that way. which will
For my
add to, like, in a statistical manner also, this has been worked out, and for verification purpose also, that may be useful something.
all the this all statistical data is already explained. Okay,
Yeah,
I
in
can
this, yeah,
add
plot way,
two
yeah,
lines.
yeah, I understand that.
I will come back to your question for that. So TNC plot is for clustering microbial community. It shows that which microbial community is present in a graphical manner. So if you go to the uh,
Why
this
that?
plot,
Yeah, yeah, these plots I wanted to understand, yeah.
yeah. So I have written the so across samples. So S11 appears in a separate cluster, which essentially means that the microbial similarity between those
Yeah,
between
looking at the
S11.
plot, if you can, speak, because this is okay, the explanation is written. Show me the plot when they are here and there, some dots are there, so how do I
So
interpret with this?
S11
Yeah.
is where orange. It's near to I think x-axis in positive side.
Hmm.
Yeah,
Yeah.
it's near to the axis. It's so what we do is in this since we already have all the data, now we compare how is the microbial clustering. So we actually it's a lot of statistical analysis for that also, but what we do is we plot values. I think in the raw data the values are also present. If you want, I can add those values here as well. I think I haven't added.
Yeah, not added.
Not added, right?
Yeah,
Okay,
so
let
that
me
is a
make
if.
a note
Yeah.
so it would be easier to visualize also, right?
Right.
Uh, Values. I think values will provide you much more clear vision.
Yeah, yeah.
I might not be able to add in the graph, but
Hmm.
at least here the
Hmm.
figure statement I can add,
Okay.
because it will be difficult to edit graph. Graphs are
Yeah.
usually come with that.
Yeah, yeah. That is
Okay, so
automated, and it's coming that way. So you
yeah,
may
yeah.
not be able to add. Yeah.
Yeah, yeah. Okay, so then for PCA and PCOA, you might also need the values, right? I think I
Yeah.
haven't added in this also.
Yeah.
Okay, let me make comments on this. Got it.
Because those positions only when they are placed here, there. So how do we interpret? So that was my.
Yeah, interpretation I
Yeah.
can of course tell, but then again, visualization might be tricky. Uh, then I think the PCO is the distance based. Uh, in this also values are not added.
Hmm.
Okay, I got it. I think I'm getting what kind of data you require. Okay, this I think alpha diversity should be in this also we have plotted all these indexes. These are different analysis we do to see where there is a microbial distribution. So how many microbes are evenness? How to explain in this?
Okay.
Uh,
So this is Gini Simpson index.
mhm.
So, like, even the names and all, is it by the name of the scientist who must have?
Yeah, 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 yeah.
Okay.
Of course, whoever
Hmm.
these are the
So, in
different
evenness focused means like the same microbes, like evenly distributed, of something that sort.
it's more about how balanced the microbial diversity is. It might differ from for different indexes. It changes the value. So evenness focused is more focused to see whether the microbiome, whatever microbial diversity is there, how balanced it is, or is it does it change, or the diversity is because we are trying to do alpha diversity.
Uh,
So under alpha diversity is different. Okay, let me start from alpha diversity. I think that will be much more. So alpha diversity is sample wise. So microbial diversity within each sample. So for that we use different indices. So indices is type of analysis, you can say. So for example, richness is number, not even number of species, number of hits we get for each species. So suppose
that means each species, if they are in large number.
yeah, number of hits.
Uh,
And evenness is for example,
evenly distributed in the system, or is it that
yeah,
way?
but I'm trying to explain the,
Huh.
for example, a single genera
Hmm.
won't be won't be how to say, a single genre might dominate.
Hmm.
So some of the genres might be more. So in the earlier indices, earlier analysis, it shows percentage. Now we are trying to see under each genera how evenly the species distribution is. So
Okay.
for that we use different analysis. So the gene assumption is based on evenness to see how balanced it is. Then inverse gene assumption is uh, evenness and number of species
But
we
here
get.
it is not written anywhere that it is for the genera. So at least add that also.
Okay, sure.
And even if the values can be, like if I'm saying richness, so in what kind of a value that I call it as richness, and if in case of evenness, how is it? Like with the help of this plot only, so that in the plot we can understand that evenness and the richness.
Ma'am, that report, okay, all of this is software generated. If I want to access that amount of data, that is huge amount of data. It won't be possible for me to go back and check every index value individually. That won't be possible.
No, that's okay. But in one, at least one or two place, like if we can.
I don't think so.
No? Okay.
The thing is, the amount of data is very big, and
Okay.
the analysis is not done by me. I'm just a person who is explaining all the data because I know this.
Okay. Okay.
But to check back and see the values,
No,
it's
because
very
when we claim or when we say that it is richness or evenness, so I have to know na that on what basis I'm saying that. So probably with the value, or how did we, like, of course with the plot it is seen, but maybe plot near to the axis or far from the axis, or what, how do I interpret it in that manner?
okay.
Like here, though we cannot see the axis also. It's shifted, yeah, on the left hand side. Yeah, one side only all.
Let me check. I don't think that's possible, but let me check.
But at least with the plot which is already there, if you can explain that.
Ma'am, all the plots are cumulative data, so
Acha.
it means that the data is huge, it's cumulated, and then the plotting is done.
Okay.
And it's all done inside of software. It's not we are using Microsoft Excel to see what values are given. And
So
all of this
what is the kind of this software which gives us?
Quine, it's Quine which uses Java and R.
It is
Quine, in the report it's written, it's a software or
QIN.
a platform. QIIME.
Acha.
And the programming language is Java or R.
Acha. Okay.
I can check. Let me ask for this if it's possible, because all of this is a huge number of data.
Yeah, because
for
looking at these plots as well as the other one, line graphs. So I from both, like I wanted to, like, how do I interpret that, if at all it is plotted, but then if I have to interpret that, how do I do that? So that was in my mind, for which I was again asking how do I interpret the graphs.
the interpretation I have written, but it's not based on the value. It's based on
Yeah.
the location, how the pattern, because I can interpret patterns very accurately.
Is
That's
that
why,
pattern is explained? Just go up.
yeah,
Let us read the
yeah.
pattern if you are
All
saying,
the
because
the final summary of alpha diversity is given highest richness. But yeah, I get what you we want to say. How will I know this is in highest richness? So
yeah,
value will help me. Okay, let me check back if I can extract that data from that file and then update this. I think for the beta diversity, you also want this, right?
yeah,
Values,
yeah,
because it's abundant space, whether it's present or absent. That's it.
yeah.
That
Okay.
is done. In this, at least, I think the coordinates are given. Let me check whether all these values can be added or not. At least for one sample, not maybe
Because
not for
normally
other.
when we do these graphs and all, automatically it takes the values
Yeah,
and it
this takes
shows.
value, but this
Huh.
is inside a software.
Huh.
So a lot of values are predetermined because it
Maybe
works on a,
values in a very large, I mean, it's like very big number, so maybe
yeah.
that it has
So
been
it's a
avoided.
lot of them is predetermined, so it's difficult. But let me check whether I can extract all these values and then add to it for different plots, at least for each species.
Like, see, no, now these graphs also, how do I, like, how do I interpret now this? Like, one brown is down, so it is S7. S7 is now, how do I interpret this?
It shows sequence per
In lower,
sample. So how many of sequences we have, and out of is the OTU table, but in a graph manner for individual sequence. In above we shows the overall, and this that the same table uh, values individual sequences is plotted as a refraction measure. So it's essentially observed OTUs. So for each point, giving values might be difficult, but let me see again if I can extract values for that.
that is why today it's become like, with the softwares and all, that
Yeah.
we have become so dependent on these things. The earlier how we used to plot and we used to know each and every point and everything. Like now,
Yeah, ma'am, but the amount of data we are generating with
very high.
metagenomics,
Yeah, yeah,
it's very,
yeah, yeah,
uh, it's a boon for at least for scientists like me,
yeah,
because
yeah.
without that we cannot analyze this
Yeah,
amount
very
of
true,
data
very
manually.
true, very true.
And then chaos index, this is species per sample. Again, it
So when
shows
the peaks are
individual.
there, suppose green peak is high. So what that green stands
One
for?
particular species is
That
high, but
S.
which one
Huh.
is there, I cannot say that.
So that is why, now the species is high, we say that in S10, green signifies for S10. So if
Yeah,
species
one
is,
of the particular species percentage is higher, but which one? That one is not.
no,
You
maybe
cannot.
we will,
It's just
maybe we will be able to, where that has been already identified, what is there in S7. So maybe looking at that, with this graph I can say that yes, this is dominant.
okay. Let me check whether I can do the values or not. I think okay. I got it. Well, you want values to interpret. Okay,
No,
this is
suppose that
the.
green is high. In the S7, we saw green is high. So
Uh,
maybe instead of data, we can write that yes, this species which is very high. Green means
Ma'am,
suppose a particular species which is very high in that particular sample.
yeah, that is written. I think this is the leptospira breakdown, because initially I was not only leptospira, but let me go down. Yeah. Okay. So after talking to you, wanted to know whether the sample risk evaluation. So I did this analysis after using that amount of data.
Uh-huh.
So that's why I color coded it.
Okay.
What species or what pathogen is there which is very high. So in this, that's why
So
you see here.
from this only, suppose S7, go to S7. What is that S7? What are the species there? Sample S7. because
Yeah.
there we saw in the peak it is very high. So that's what I was trying to say, that in this, suppose I get S7, what it stands for, see here, suppose it's
It's
pseudomonas.
Pseudomonas.
Ha,
The
ha,
peak might be for Pseudomonas,
ha. So that's what.
but there is no way to know correlate
Ha,
that
ha. But then
particular
see,
graph
ha,
to.
because see, if you see the value, that is 0.2 percentage. And still I see the peak very high, whereas pseudomonas 3.4, see in S10 it is 3.4%.
Yeah,
Got my
the
point?
graph is not based on percentage. The graph is based on
The species which
sequences.
is very dominant.
Its number of hits.
Ha.
Yeah, its number of hits rather than percentage. It,
Number of hits means again and again reoccurring, so means like repeatedly
stable
that
occurring.
species.
Yeah, yeah.
But then if suppose in percentage, acha, okay. Okay, percentage you are saying we are not counting.
That's why the different there is different graph for different analysis. So if we go here quickly,
See, this only I was talking, that peak is, you can see that S10, it stands for S10.
yeah,
Okay, yeah, S10 was high, right.
yeah.
So in that species, Pseudomonas species, it was high. So maybe
No, I think S10 it's the highest is leptospira. If I'm not wrong, let me check. I'm not sure.
go up, go up. Acha, okay,
Uh,
S10, go to S10, then we'll come to know. Yeah, see S10.
yeah, Pseudomonas and Enterobacter.
Yeah, I said,
Yeah,
I saw that
common in
is in highest,
environmental
highest
samples.
percentage it's showing. So maybe it
Yeah,
is that.
2.5%. Yeah,
Because in other cases, Pseudomonas is less, percentage wise also when I
less.
see.
Yeah, it depends. And the last column is not last column. The second last column is potential risk and implication, and the last column is
Causes.
possibly the best of my knowledge.
Hmm.
And then for overall risk summary, I have given high risk, moderate risk, low, and the last is action plan. It's basically a literature review.
Hmm.
I didn't
Hmm.
do much. I
Hmm.
have to just see if
But
it's
then,
high risk.
in this, like in this interpretation and action plan, that can we also mention that these are the good bacteria which are there, which can be maintained at so and so, so that will be helpful to revive the water body, and maybe that these are the bad bacteria, if they are worked on that, and they are like killed, then only things will change, of that sort, something.
Okay. I can do that. Let me. I have to just check what kind of good bacteria are there. I rarely found any, but okay. I can do that.
Because
I just need to.
that is the major requirement of this particular testing, because we want to maintain good bacteria, we want to destroy the bad bacteria like pathogens which spreading pathogens and all, and in combination probably if there are fungi or some viruses there, that they are more becoming
With
virulent of that
16
sort.
is we can't identify fungi and other stuff, but
I know that, but
I can check for what are the bacteria which are less harmful
hmm,
and can be balanced out
yeah,
to improve the risk assessment.
yeah.
Okay, okay, got it. I will make another table and add below it.
Because mainly, in the riverine system which is continuously flowing, and one which is like suppose a pool or this kind of a lake which is closed system, they're using some bacteria for treatment, and in a flowing system it's totally different. So in that sense, that how it can work of that, something of that sort also was like I was looking for. Just a minute, hold
Uh,
on, somebody has come. I'll
yeah, very proficient in this area, but I can make minimal suggestions based on my experience.
hmm.
I can provide a list, but other than that, I might not be very
No,
helpful
it's
in
okay. It's okay.
providing
That I
suggestions.
will work out on that, because that will be maybe through the references and all only I'll be able to. But
Yeah, I can make another table to say what is the abundance of
yeah,
bacteria which are non-pathogenic and
yeah,
how can they provide help.
yeah, that would be helpful.
Okay. Let me redo the report. I'm
Yeah.
actually flying out on Saturday. So
Okay.
I might not be available for 2-3 days, but I will reach out to you next week.
Sure, sure. Okay. Even I am in Nepal. I will be also flying day after. So
Okay,
after
okay,
two
sure,
three
ma'am.
days only I'll be coming. Yeah. I'm going to
Okay,
get some
okay.
samples from there also.